I emailed you that. Did you see what I made? So it's like, you should, Natalie said I would do it. No, that was the greatest one. So is that a whole different boat? That is from earlier? Yeah. Okay, because I was trying to say that. And I was like, I already have that use for that. It's been sad. You did that all.
For lab tomorrow, we are doing one stain, the acid-fast stain. You do not need a culture of a particular age for that, and so as long as you have some viable growth on the slants, or even a broth for that matter, you can do that acid-fast stain. Kind of a messy stain, so don't worry anything you value. And gloves probably will be in order tomorrow. Okay, I'd like to finish up the last stuff that's going to be fair games for the exam on Monday. That will just take a few minutes, and then I will stop and see if you have any questions. And if not, we will.
Whatever, I'll ask you when you have questions.
Okay. I'll say a little bit about the exam too.
That's what I was asking.
Okay. So let's finish our slog through Berkey's manual. We're almost done. We are now in volume 4, which are the high GC content gram-positive bacteria, as opposed to the low GC content bacteria. And yes, I'm recording. Then we're in volume 3. Taxonomically pretty easy, single phylum, divided into one class. I want to just briefly mention a few of the genera within that single class. One genera is the genus Actinomyces. These are oftentimes referred to as the branched bacteria. You ever looked at fungi under a microscope? Fungi are, even the macroscopic ones like mushrooms, are actually made of little filaments called hyphae. And those filaments intertwine with one another and can form a pseudomass of tissue. These bacteria are oftentimes very fungus-like in appearance because they have this branched, hyphae-like looking appearance. Really abundant in soil. And that's where they play one of their major ecological roles. They are very good decomposers, including the ability to degrade cellulose. So they are a recycler of dead plant material. You ever notice when you pick up soil and it's got kind of a musty smell to it sometimes? That is due to primarily those Actinomyces bacteria. And another significance for these is that many of the species within this genera produce antibiotics. So when we talk about antibiotics, we'll come back and kind of mention these guys again. That's why I threw them out there. Another genus that has some significance is the genus Mycobacterium. And this will be the primary thing you're going to be working with on Friday when we do the acid-fast stain. When we talked about cell wall structure, we said that some bacteria particularly bacteria associated with this genus, had this waxy substance in its cell wall called mycolic acid, and that made it very difficult to stain. In the acid-fast staining procedure we use tomorrow, you will put some growth on the surface of the slide, and then you'll put a paper towel on top of it and soak it with stain. And then you'll actually invert your Bunsen burner and you'll direct the flame down on that soaked paper towel, and that will cause the stain to steam, and that will force the stain through those waxy cell walls so that you can stain the cells. Otherwise you can't stain them. We will then do a decolorization, and so anything that's not acid-fast, in the decolorizing process, that red stain will be washed out of it, but the acid-fast bacteria will retain the red stain. And so you can see a difference after the acid-fast staining. The acid-fast positive are red from the primary stain that was applied, and then we do a counter stain of methylene blue for the ones that were destained. So it's a little bit of a messy stain, and you've got to really work to get that stain into the cells. We'll do something similar next week when we do the endospore stain, because it's also hard to get stain into endospores to stain. So we'll use that same steaming technique. Okay. We want to use the older Bunsen burners for this exercise because they give us that longer kind of cone of flame. Those newer Bunsen burners only give us that really short thing, and it really doesn't work very well for the steam. Okay. What I would recommend that you do is that you work with your partner. You'll have your unknown, and then you'll have a mixture of some acid-fast, non-acid-fast bacteria on another slide, and then you can kind of stain them and steam them all at once, you and your partner can. Okay? So these are acid-fast positive bacteria due to the presence of the mycolic acid, the waxy substance in the cell wall. Here is some clinical importance of a couple of species in the genus Mycobacterium. This particular species is the species tuberculosis. One of the things that the body oftentimes does in response to a TB infection is it walls off the bacteria in an attempt to try to localize them and keep them from spreading. And they can form this kind of thick wall around them called a tubercle. And in the lungs on this particular x-ray, you can see some tubercles, these light staining areas right here, that are very, very characteristic of a tuberculosis infection. TB used to be a real scourge around the world. And even in the 1930s in the United States, there were a lot of cases of TB. There wasn't really any good treatment for it. There was some belief that sending people to the western purer air or Rocky Mountains was helpful for the breathing difficulties associated with tuberculosis. And so they developed various sanatoriums where people could go for TB treatment. Once antibiotics were discovered, TB really came under control pretty quickly. And so TB really kind of became a disease of the past. Now it's making a reemergence, in part due to the fact that there are TB bacteria that are developing multi-drug resistance. And so TB is becoming an increasing public health concern. The treatment for TB, the antibiotic treatment, is a very long treatment. Mycobacterium grow really slow, and the slower that an organism grows, the less susceptible it is to antibiotic. So it is not uncommon to have to be on an extended year-long antibiotic treatment if you are diagnosed with TB. Y'all had a tuberculin test? Y'all done this before? Little scratch test. Actually, this particular test is determining whether you have a prior or current infection of TB. And what they do is they inoculate your skin with a little bit of TB antigen, and then they look to see if you developed an immune response against it. It's kind of like, philosophically, the scratch test you do for allergies. But for allergies, you get a pretty immediate reaction. Allergies are called immediate hypersensitivity. TB is a little bit different mechanism of immune response, and it causes a delayed hypersensitive reaction if you've had prior exposure. And so it can take a couple of days for that little bump to develop that indicates delayed hypersensitivity. So this is the basis of your TB test. Okay. Another infamous disease caused by Mycobacterium, but a different species, is Mycobacterium leprae. And this is the causing agent of leprosy. Leprosy is a very slow progressing disease, again, because the mycobacterium grow really slowly. And these particular bacteria have a fairly low temperature optimum. And that's why the places that are most affected with TB infections are the extremities, because your extremities tend to be a little bit colder than your core body temperature. So face, hands, feet are where you will commonly see leprosy manifest itself. And it can result in necrosis of the tissue, and you've probably heard of situations where, like fingers and toes literally can fall off of people who suffer from leprosy. What's really kind of ironic about leprosy, however, is it's really not very contagious. So the practice that's been in place since biblical times, and really up until the 1800s when there was a leper colony on Molokini in Hawaii, that idea of segregating those leprosy people because you're afraid that they would cause infections turned out to be pretty much unfounded. Okay? It's really hard to get leprosy from casual contact. You really have to be in pretty intimate contact with people. So another pretty nasty disease. And then a final genus within this phylum of volume 4 is the genus Micrococcus. This used to be grouped in previous edition of Bergey's Manual with the Staphylococcus and Streptococcus. Micrococcus, as the name indicates, are cocci. Most commonly, they form little packets of 4 called tetrads. Remember, the strep were the chains, the staph were the irregular clusters. Micrococcus tends to be mainly tetrads. So at one time, they were all grouped together, kind of taxonomically. But now when we're doing genetic comparisons, they really fell in a very different phylogenetic group. So we'll do some work with Micrococcus. We'll use Micrococcus luteus, for example, pretty routinely in lab.
Is the leprae, whatever, is that species or a genus?
That is a species within the genus Mycobacterium. Okay, so Mycobacterium tuberculosis is one species, leprae is another species. Okay. And then finally, volume 5. Volume 5 is kind of a mismatch of a lot of different types of organisms. Planctomycetes, Spiracades, Fibrobacters, Bacteroides, Fusobacterium. Several phyla. There's really only 2 phyla that I want to briefly mention right now, and that is the second and third phyla down here, the phyla Chlamydia and the phyla Spiracetes. This is volume 5. phylum Chlamydia. Chlamydia bacteria are also obligate intracellular parasites. What one did we talk about last time? It was an obligate intracellular parasite. Remember? Rickettsia. Good. Rocky Mountain spotted fever bacterium. Little small, tiny bacterium. This one's also small. Actually smaller than Rickettsia. But unlike Rickettsia, it is not transmitted by arthropods, so you don't have to get a tick bite, for example, to get chlamydia. The reason that these are obligate intercellular parasites is that they can't make ATP. So they really are energy parasites. So they've got to be living inside of another cell can manufacture the ATP for them. Chlamydial infections are one of the most common sexually transmitted diseases, but they are oftentimes undiagnosed because they don't have the overt symptoms that syphilis and gonorrhea have, for example. We're now coming to understand that chlamydial infections may be a major cause of infertility in women. The chlamydial infections can scar the fallopian tubes, for example, and make it difficult for the eggs released from ovulation to then get into the uterus to implant. Chlamydial infections can also be spread to a newborn during the birthing process. And one of the places that infants are most vulnerable is the eyes, because they have very little protective mechanisms there. So chlamydial eye infections, if they're not treated, can cause permanent blindness, for example. The old treatment used to be a prophylactic application to a newborn immediately following birth of silver nitrate drops in the eyes. Because one of the things we'll talk about later on when we talk about controlling growth of microorganisms is a lot of metals, including silver, are antibacterial. Anybody ever gotten silver nitrate on their skin? Yeah, it turns your skin kind of brown and it does burn. So these silver nitrate drops were really not very pleasant, but it's better than having your newborn child get an eye infection that could lead to blindness, particularly if you were undiagnosed. Now they pretty much replace that treatment with antibiotic drops. Erythromycin, for example, is used pretty routinely. So newborns are just prophylactically treated with these eye drops in case they might have an undiagnosed chlamydia infection. So these are the little tiny chlamydia bacteria, very, very small. Give you an idea of size. These are the little microvilli that are on intestinal cells. Microvilli are just little individual membrane projections of an epithelial cell in the intestine. So these chlamydia are really, really tiny. The other phylum in this volume that's worth mentioning is the phylum Spirochetes. These bacteria are associated with a wavy helical type of morphology. You all saw a fairly large spirochete in lab when we looked at the Bacillus cocci and Spirillum bacteria. These bacteria oftentimes have that endoflagella. You may or may not remember that an endoflagella wraps around the cell. And when the flagella move, It causes the cells to move in a spiral, kind of corkscrew fashion. And they can really then burrow into tissues. That makes them hard to get rid of. One of the more infamous spirochetes is found in the genus Treponema. And particularly the species Palladium, Treponema palladium. This is a causative agent of syphilis. Syphilis has been around pretty much as long as mankind has been around. It is transmitted primarily venereal, it's a sexually transmitted disease. And up until the 1940s, when antibiotics were developed, this disease was a gargantuan public health problem because there really was no treatment. And since people didn't necessarily understand the cause and effect relationship and how they were getting it, it was very hard to tell people to alter behavior to try to prevent getting this. Okay. Syphilis, this is an eye infection from a chlamydial eye infection, pretty nasty. And this is the application where they're getting ready to do eye drops from newborn infants right here. So this is a spirochete bacteria like syphilis, corkscrew, spiral shaped, burrows into tissues. Syphilis goes through an interesting progression over a long period of time, referred to as the primary, secondary, and tertiary stages. In primary syphilis, usually a blister-like lesion erupts, oftentimes on the genitalia, could be on the lips and mouth as well. And that particular lesion is called a chancre. It doesn't last that long. And if the chancre disappears, a lot of times people believe that the syphilis had then resolved itself. It magically disappeared. But really, the bacteria just went into a dormancy or a latency, and oftentimes would then manifest itself later as secondary syphilis. And that oftentimes is associated with a whole body rash. Very painful. Very obvious in this particular case. But again, that oftentimes would resolve itself as well and would disappear. So again, people would say, well, okay. The third stage, which is usually lethal, is associated with tissue degeneration. So we start getting necrosis of tissues. Here is a tertiary syphilis victim, and you can literally see part of the nose here being eaten away from that tertiary syphilis. Again, once antibiotics were developed, syphilis kind of became a non-concern until multi-drug resistant forms of syphilis started showing up. Fortunately, now though, we do know about the causative agents of syphilis, and people can make some choices about being tested, being with partners who've been tested, abstinence, condoms. You know, there are some other ways you can deal with this that weren't available when we didn't know what the causative agent was. And finally, one final genera, that is the genus Borrelia. species Burgdorfii. This is the causative agent of Lyme disease. You probably all have heard of Lyme disease before. So here are the spirochete bacteria right here. Borrelia burgdorfii. And one of the things that oftentimes is associated with Lyme disease is this rash that has kind of a bullseye appearance. And so there are kind of concentric circles of rashes that looks like a bullseye. But it's oftentimes not as dramatic as this is. If you are diagnosed, antibody treatment is very effective. But a lot of times people really aren't diagnosed, the rash dies away, and you really don't know you had it. You might have some flu-like symptoms or something. But then oftentimes this manifests itself later with some severe arthritis, and so really tremendous pain in the joints. And by that time, it's really hard to treat. How do you get Lyme disease? Infected ticks, okay? The Lyme disease ticks tend to associate with a particular species of deer called the white-tailed deer. The white-tailed deer is infected, but it doesn't really show symptoms. The tick bites it, picks up the bacteria, bites a human, infects the bacterium. The human isn't affected, shows symptoms. Lyme disease used to primarily be associated with only the northeast part of the United States, New Hampshire, Vermont, that area. But it has been spreading over the years. Indiana now, it's not uncommon for Lyme disease cases to be shown. Why is it spreading? Yeah, the range of the white-tailed deer has been expanding, in part due to climate change, in part due to urbanization. And those deer have been migrating out, and where you didn't used to find white-tailed deer in Indiana that much, they are becoming much more common now. So the range of that disease is spreading as the range of that host organism has spread as well. Okay, so that completes our run-through Burgess Manual. As I said, at the very end, there's this slide that kind of tells you what kind of the bottom line is in terms of what you need to know taxonomically for these organisms that we've gone through. Okay? So you need to understand basic structure of Burgess second edition. But then as we talked about specific examples, you should be able to go back and relate those to a volume, a phyla, and then if we talk about genus within that phyla, that as well. In some of those instances where there are long lists of phyla that we didn't talk about, for example, in volume 5, we only talked about 2 of the phyla. Those are the only ones I expect you will know. Okay? So you don't have to know the names of all of these other phyla that we didn't cover. Same thing over here in the first volume. Okay? There are only a couple of the phyla that we talked about. All the rest of them, don't worry about having to know the names of all those. So it's really more manageable. As far as the exam is concerned, since many of you have not had an exam for me, I would also say in the past, well, they're pretty much like cell biology exams. Typically, about half of the exam is subjective, and those are multiple choice questions. So typically, I'll have somewhere between around 25 multiple choice questions. I do not do true false. I do not do matching. I do not do fill in the blank. The rest of it is objective, short answer, and essay questions. One of the essay questions, short answer, that I love to do are agree, disagree questions. I think other faculty have kind of picked up on those too. Have you had those before? Todd is, because he said me. In agree, disagree, it's typically like a 4-point question. You are given a statement, and then you're asked to agree or disagree with it. But then you're asked to justify your position. So in some respects, it's kind of almost like a true-false, but with explanation. Okay? And the key is how you justify your position. It is not uncommon for me to give full credit to someone who agreed with the statement and full credit to someone else who disagreed with the statement because of the way they interpret that statement and the spin they put on it. Okay? So really the big thing is justifying your position. And if you look at old exams, you will see examples of agree and disagree questions. There are then probably going to be a couple of a little bit longer essays. My essays tend to be 8 to 10 points max. Okay. So if you look at old exams, you can kind of get an idea of the structure of the exam and how many questions there are. I do make up new exams each time, so don't just try to memorize a bunch of old exams. But I will be honest with you, I mean, there are only kind of so many questions that I can come up with, and so it's not uncommon for you to see questions on the exam we'll take that might have been asked in the past. It is not uncommon for essay questions to come from the study guides. It is not uncommon for a study guide, what was an essay question, to become a multiple choice question. So I really would urge you to do the study guides. And they tend to follow the notes, okay? The emphasis here is lecture notes and PowerPoint slides. You really should only be going back to the book if you need clarification. Okay? For the taxonomy, up until last year, I was using primarily the taxonomy from Berge's first edition of Systematics. So there's a lot of questions about Berge's first edition and really nothing about second. Last year, I switched over and started emphasizing Berge's second. So I want you to be aware of that when you look at old exams. The way the taxonomy section looks on last year's exam is kind of different from the emphasis on previous years. Other thing is that we didn't get quite as far this year as I've typically gotten. So I think last year there were a few questions about growth of microorganisms. And so if you see material on an old exam about growth or something that you don't understand, we didn't cover it yet. That's going to be on the second exam.
For the Berge's volumes, when you showed us all of them, but then only explained like a few important ones.
Yeah.
We just need to know the few important
Yes.
ones and their names.
Yep.
We don't need to know the other ones.
Right. Yeah. Now, I mean, you should know the kind of general characteristics. So you should know volume, 3 is, this group of organisms. Volume, 4 is the low GC, okay. But in terms especially of the names of the phyla and stuff, so I'd really like you to go back and kind of take the examples and fit them back into the taxonomy of those, okay? So if we didn't talk about, several of the phyla within one volume, don't worry about it. So what questions did you have about? You said questions about the exam?
You answer them.
I answer them?
Yep.
Any questions about exam material? You've got basically your introduction, history, you've got your bacterial structure, and we've got the classification. Yes, sir.
You said the dates were purely aesthetic, right? We don't need to know the dates for history.
You don't need to memorize the dates. I think it's important to understand contextually general time frames. Okay? So, microbiology did not develop as a true science in the 1500s. It was the 1800s. Okay? Names, I do like you to know names because I think knowing the names of people that develop the science is important. The dates. I feel the same way about dates as I do chemical structures and memorizing them. You can look them up. If you have a general idea, that's good enough. Nicole, did you have a question?
Yeah, I was looking at one of the old, this is kind of a stupid question, but one of the old guys, and I saw your multiple choices go linear instead of horizontal.
To save space, yeah.
Is that gonna be that all? I just, when I read, sometimes I to miss the line. I'm done at least.
I mean, I suppose I could, but it's gonna take pages and pages for the exam.
That's fine.
Yeah, I mean, that's just kind of the way I've always done my multiple choice.
That's fine.
Oh,
Going back to the history part,
Mhm.
I was doing the study guide, and one of the questions discussed the history of development into the microscope. And all I really have in my notes is about Leeuwenhoek using it for cloth and everything like that. Is there more we need to know about it? And I know, then you said the development of the electron microscope is prokaryotic and eukaryotic, but
yeah, I mean, the first microscope was really just a single lens microscope. And so part of the development of the microscope was going from a single lens to a two lens microscope, the compound microscope. And there you get the advantage of the multiplicative factor of ocular and objective. And I guess then the next real era in development was the development of the electron microscope. And that's when we really saw this new world of internal structure and realized that there were, prokaryotic, eukaryotic cell structures. Those are the major elements. Got it.
I think in class you just mentioned it, but can you go over the medium and
Some years I've spent a little bit of time, kind of talking about that in more detail. But basically what happened is that Needham was a fierce proponent of spontaneous generation. And so he would do an experiment that he thought would support spontaneous generation. And then Spallanzani was a fierce proponent of biogenesis, life coming from life. And so he would do another experiment and say, no, Needham's wrong, I'm right. And then Needham would come back and do another experiment and say, well, in this experiment, I'm right, he's wrong. And one of the things that Needham was famous for was these kind of outrageous assumptions. And so he's the one that came up with the criticism that, well, when Spallanzani's boiling his flasks, he's injuring the air and destroying the vital process in the air that causes spontaneous generation. So Spallanzani goes, how do I deal with this? And so he developed this flask then that was actually open to the air, so fresh air could get in. So that kind of back and forth, they kind of illustrate the tension between kind of religious-based science, because the religious people really didn't want to give up on spontaneous generation, and the more science-based scientists who were constantly counteracting the ones that really believed in that it didn't occur. So that's more than enough. Sometimes I'll talk a little bit more about their individual stories and stuff, but I just did this stuff. Yes?
There's a question that says about the aseptic and pure poultry techniques and how they contributed to microbiology. It just gives, those are helpful to make it possible to test these theories that we have and whatnot, right?
Well, those techniques, aseptic technique which prevents contamination, and then pure culture which allows isolation, are absolutely necessary to generate pure cultures of microorganisms. So that's the only way you can be sure that you're working with a single individual species. And if you don't have a pure culture, if you have a mixed culture, you've got an invalid scientific experiment because you've got more than one variable. So they really, were necessary to be able to do controlled experiments in microbiology. And if you can't do a controlled experiment, you're not a scientist.
we were talking about, I think that it was the question that said something about like the industrial uses of bacteria or something. We talked about gunpowder.
That was just kind of one little story. There were several slides in there that just gave you some, we talked about the food industry, for example, and, especially dairy products. But then we also mentioned that there were a number of, acids and other chemicals that were produced originally by microbial fermentation and now are being produced more by organic chemistry using petroleum products. We mentioned a little bit about antibiotics. That's another industrial kind of application of microbial metabolism. So some of those slides that were in there are just kind of, look at those, they're kind of illustrative of the diversity of uses that microorganisms have industrially. And sometimes I'll throw in little stories. I mean, I don't think I mentioned indigo, which is used to dye blue jeans, can be produced by microbial fermentation. The gunpowder story, there's, you know, a million stories out there about uses of microorganisms for industrial processes. And then there's the whole biotech industry. So, very important. Yes.
What's Vibrio? What's the adjective about morphology? V-I-B-R.
Oh, Vibrio. Vibrio. It is a wavy form of helical bacteria as opposed to the spiral one is more twisted. Vibrio is just kind of a wavy form. That was probably on one of the slides, wasn't it? Yeah. Oftentimes Vibrio even have just a single little bend in them, so sometimes they look almost like a comma as opposed to just a little short nod. Yeah. I will let you come in early if you want to get an early start on the exam, especially if you have 9 o'clock classes. But it is designed to be an hour-long exam. If you just want to put a little bit less pressure on yourself, that's okay. Other questions? I mean, let's say 8:30. Okay. I mean, I normally have the exam ready to go at 10 till anyway, so that's only an extra 20 minutes or so, but sometimes extra 20 minutes is helpful. So tomorrow I have some fast stain. While that's drying and stuff, you might be able to kind of look at some of the other slides that we did Wednesday if you haven't had a chance. We'll work on some contamination issues we might have with a couple of cultures. So, well, I'm not going to start anything in 5 more minutes, so we will pick up next Tuesday with a new topic. Oh, okay.
I knew that it was like with the but I'm
Yes. I'm not sure that we know absolutely what the purpose is. We know it's there. We know that it is a negatively charged molecule and it contributes to the overall negative charge of the cell. Or whether that's I mean, there must be some function for it.
yeah,
But,
it's not something we know yet.
right. Yep. But you should definitely know the teichoic acid is associated with the peptidoglycan layer,
Yeah.
and primarily the peptidoglycan transphosphorus.
And then what coming on this one was the exception to the alternative.
Well, I mean, the ones that lack a cell wall, okay?
Oh, okay.
And then there are the archaea bacteria, which have cell walls, but they're not peptidoglycan.
Right.
And then there are also the L forms, where sometimes they have it, and sometimes they don't have a cell wall. Yeah, definitely right.
Did you say that we needed a broth culture or unknown?
No, you can have a slant or broth.
That's okay.
Yep.
All right, and you said that you would check to see if our gram stain was correct with our unknown.
Yeah, if you would, just give me a piece of paper and put the number of your unknown, cell morphology, and the gram reaction, and I'll go look it up and
Okay.
get that back to you.
Thanks.
Do we need to have you check it if it was like really obvious?
I would have me check it.
Okay. It was unmistakable.
If you're going to get free information, get it.
I was happy that it was a mistake.
So it looks like help got onto that computer now that we rebooted it.
Yeah, it works, but I don't think it's going to be
It is now again. Yeah, we got it resolved. So.
You can print in here for free.
Yep.
So
And actually, if you got charged supposedly for printing in here, your account's being refunded. This is 210. They can't print in color.
Did you say you're trying to print on the internet?
well, it could cost me about.
It says error, one document.
Clear jam.
Oh, look, it says, I needed this as a policy. I learned just this. Is it printing or not?
Okay, it's printing something right now.
Oh,
No, it's not. It says jam and tray 2. Maybe that is tray 2.
Oh, I just think it. Is it different ones?
Maybe not.
Dr. Breder, for my negative stain, I mean, my culture is 48 hours old. That'll be fine, right?
Yeah.
Okay.
Negative stain doesn't matter.
That's what I figured.
No, somebody's. See, people printed this and then don't realize they sent it to the wrong thing.
What do you try to print, Audrey?
I didn't try.
Okay. It says it's ready now. Let's make this.
You can try to re-release it.
Okay, give that a try.
Here's my sheet.
Oh, okay, thank you.
Thank you.
Is this the only one we can print for free?
I think in any of the science labs, these kind of printers you can print
Those
for free.
ones?
Yep.
'Cause I don't think those can print double-sided, can they?
Probably not.
A lot of them are on the fritz right now, though. Especially on the some of the chemistry ones. I was trying to talk to you last week. We could not get printers to work.
Yeah, I know they're working on it.
Our IT department makes me sad sometimes.
Are you waiting for me, Eric? Bye.
If we give you what we think our morphology and grade stain was and it's wrong, we get it tried again.
Oh, yeah,
Right?
yeah.
Okay.
Yep.
I didn't know if it was one of those things where it was like wrong.
You good?
Good. How are you?
Okay.
So Jeremy rebuilt this computer. I'm guessing replaced this one.
Yeah, this one, it takes like half an hour to load PowerPoint.
So, okay. So I know Jeremy mentioned you had some files on here that you wanted copied off.
It's really not that big a deal. I've got it.
Vintage one?
I just put it on like that copy to your app.
Oh, okay.
So don't worry about it.
Okay. Yeah, I was gonna say
It'll
otherwise.
take forever to transfer this computer.
I was gonna say I was gonna ask. Oh well, the files on there. Would you want me to put them on your G drive? They're already there.
Yeah. So.
No.
So yeah, if you wanted to swap this one out with that one.
Yeah. I think that'll work. It's also a little bit smaller too. So.
Yeah, that'd be nice. Yeah, it's funny, those computers, I mean, last year it was slow, and then this year it was just
Oh, I'm
to
sure.
the point where it was just unusable.
The hard drive's probably shot on it then. Is that a VMware or no? It's actually just.
Yeah.
That's stuff.
Yeah, and I have yet to hear back from Tensig about the thin clients you have over here. They're supposedly still working on fixes for things.
Okay.
So,
I mean, what I'm still finding is that pretty much each day I just need to reboot because if you come in here and they have to log in screen, if it's been sitting for more than about a day.
yeah.
They just don't log in.
Well, I know yours are the slightly older versions compared to what we have like over the library. I know the library ones get a lot of use. But yeah, the company that makes those right now, they're on my list for
Yeah, it is.
frustrations.
Huh.